So, if you've paid any attention to the whole regenerative medicine boom lately, especially with the sheer volume of new therapies hitting the market as of August 2026, you've probably noticed a pretty stark contrast.
Oh, absolutely. It's impossible to miss if you're in the field.
Right. Because on the surface, these high-end medical clinics project this image of, total pristine control. They promise the literal future of healing. But behind those clinic doors, there's like a hidden, really high-stakes battle playing out right now. And it's actually causing entire practices to just quietly shut down.
Yeah, and the crazy part is it has almost nothing to do with the medicine itself failing. It's entirely about how global governments classify it.
Exactly. And that's what we're getting into today.
It is a massive regulatory bottleneck.
Right.
Because what looks like a simple, futuristic injection to a patient is actually the center of a really profound scientific debate. Like what constitutes a drug? What constitutes a tissue? And, well, who gets to decide?
And that is our exact mission for this deep dive. We're sorting through a pretty dense stack of sources today. We've got FDA warning letters, European Medicines Agency guidelines, biotech research papers detailing manufacturing standards, and even risk assessments from liability insurance firms.
Which are terrifying to read, by the way.
Oh, truly. We're going to decode how global agencies, specifically the US FDA and the UK's MHRA, classify living human cell therapies versus cell-free extracellular vesicle products.
Right.
So whether you are evaluating a cutting-edge aesthetic treatment for yourself, or you're studying biotech, or you just really want to understand the rules governing the future of human medicine, this is basically your shortcut to understanding a very complex game.
Yeah, and to really grasp the legal frameworks here, we have to start with the physical biology.
Yeah.
Because regulators are trying to govern two fundamentally different biological materials, and the law hinges entirely on their physical structure.
Okay, lay it out for us.
Yeah. So the first category is human cells and tissues. In the regulatory world, you'll see these abbreviated as HCTPs. The classic example you'll see in the clinical literature is mesenchymal stem cells.
Wait, let's pause right there, because mesenchymal is one of those words you see plastered on clinic brochures everywhere, but it's rarely actually explained. What does that mean in a practical sense?
That's a crucial distinction to make. So mesenchymal refers to multipotent stromal cells. In plain terms, these are just adult stem cells found in connective tissue.
Like bone marrow.
Exactly. Bone marrow, fat, umbilical cord tissue. And they are multipotent, meaning they have the ability to differentiate into a variety of cell types, like bone cells or cartilage or fat cells.
Got it.
But the key takeaway for our purposes is that these are intact, living entities. They have a nucleus, they metabolize energy, they can divide and self-replicate. They're essentially the living biological engines of the body.
Okay, so they are fully operational factories,
Hmm.
but looking at our sources, they focus really heavily on a second category that regulators treat entirely differently, which are extracellular vesicles, or EVs. And these aren't cells at all, right?
Now, correct. EVs are cell-free. They're tiny, lipid-bound particles that are secreted by those living cellular engines we just talked about.
Oh, I see.
Yeah, and because they don't have a functional nucleus, they cannot divide or self-replicate. They're essentially little biological bubbles floating through the body. And they come in different classifications, based primarily on their size.
How small are we talking?
Well, you have microvesicles, which range from 100 to 1,000 nanometers. Then you have apoptotic bodies, which are much larger, like 1,000 to 5,000 nanometers.
And look at those.
Those are essentially the fragmented debris released when a cell undergoes programmed death. And finally, you have exosomes, which are unimaginably small. They range from just 30 to 150 nanometers.
Okay, just to put that in perspective for you listening, a single strand of human hair is about 80,000 to 100,000 nanometers wide. So a 30 nanometer exosome is, it's practically a ghost.
Right, it's incredibly tiny.
But if they're just these inert... Okay, that makes perfect sense. So if a living stem cell is the biological factory, the exosome is like the tiny FedEx package that the factory ships out, carrying the actual instructions for the rest of the body.
Yes. That analogy holds up incredibly well. And it brings us right to the heart of the regulatory crisis.
How so?
Because a living factory can grow, mutate, or produce unpredictable things if you place it in the wrong environment. But a FedEx package just delivers whatever was put inside it. Because one is alive and one is just in a cellular delivery vehicle, the law attempts to regulate them using completely different paradigms.
Which brings us to the U.S. framework. Reading through the FDA warning letters from our sources, it seems like the survival of any regenerative clinic in America really comes down to two specific sections of the Public Health Service Act, Section 361 and Section 351.
Yeah, those two numbers dictate absolutely everything. The Section 361 pathway is designed for low-risk human tissues and cells. And to qualify for this pathway, a product has to meet extremely strict criteria.
Like what?
The two most critical requirements are that the biological material must be minimally manipulated and it must be intended for homologous use.
Okay, just to clarify for everyone, homologous use means the tissue performs the exact same basic function in the recipient as it did in the donor. So like taking a skin graft from my thigh to repair a burn on my arm?
Yes, skin replacing skin, or taking bone from one area to fuse a spine. And the reason clinics desperately want their products classified under Section 361 is because it does not require FDA pre-market approval.
Wait, really? No approval at all?
None. You don't need clinical trials. You just register your facility with the FDA, follow standard tissue banking guidelines to prevent communicable diseases, and you can operate.
Okay, but the moment you step outside those boundaries, say you manipulate the tissue too much, you get bumped into the Section 351 pathway.
And that is a completely different universe. Section 351 classifies the product as a biologic drug. Getting a 351 product to market requires a biologics license application, or a BLA.
Which is a massive undertaking.
Huge. It means conducting investigational new drug trials, so Phase 1, Phase 2, and Phase 3 human clinical trials. It requires proving safety and efficacy and manufacturing under intense pharmaceutical-grade sterile conditions called CGMP.
Yeah.
We are talking about a process that costs tens of millions of dollars and takes 5 to 10 years.
See, I want to push back on the FDA's logic here for a second, because this is where the industry seems to be constantly fighting them. If a doctor takes fat cells from a patient's abdomen, isolates the stem cells, and then puts them in a petri dish with some nutrients to grow a larger, more effective dose...
Right, cell expansion.
Yeah. Why does the FDA suddenly view that as manufacturing a drug? The clinic would argue, hey, we're just growing the patient's own natural cells.
I get that argument, but the FDA's position is rooted in the concept that the process is the product.
Yeah.
The environment inside a plastic petri dish in a lab is radically different from the environment inside the human body.
Okay, fair.
When you force cells to expand and divide outside their natural habitat, you alter their biological characteristics.
Yeah.
You introduce the risk of genomic instability. The cells might lose their natural surface markers, or they could suffer genetic mutations that turn them tumorigenic.
So they could literally cause cancer.
Exactly. By culturing them, you are just multiplying them. You are fundamentally changing their nature. Therefore, the FDA says it is no longer just your tissue. It is an unproven biological drug.
Okay, the distinction between 361 and 351 makes total sense for living cells. If you grow them, it's a 351 drug. If you just move them, it's a 351 tissue. So why are FDA warning letters surging right now? I mean, we're seeing clinics getting raided and shut down for products they claim don't even have cells in them.
You've just identified the exosome trap.
The FedEx packages.
Right. Clinics recognized that the 351 biologic pathway was too expensive and too difficult for living stem cells. So they pivoted. They started heavily marketing exosomes, claiming that because they don't contain a nucleus, there's no risk of rogue cellular growth or tumors.
And so they argued, what?
They argued that these cellular products should just default to the simpler, low-risk 361 tissue pathway.
Honestly, on a surface level, that argument almost sounds reasonable. If they don't have DNA that can mutate and form a tumor, why regulate them like a dangerous drug?
Well, the FDA has issued incredibly blunt safety notifications addressing that exact argument, and they reject it on two distinct scientific fronts. First, look at how exosomes are harvested.
Okay, how are they?
A lab has to culture stem cells, collect the liquid media those cells were growing in, and then use complex ultracentrifugation.
That's spinning it really fast, right?
Exactly, spinning the liquid at massive speeds, or using advanced chemical filtration to isolate those tiny 30-nanometer vesicles. The FDA states definitively that this isolation process alters the biological characteristics of the starting material. It is, by definition, more than minimal manipulation.
Oh, so it fails the 361 test immediately. And what's the second front?
It goes back to the very definition of the law. The 361 pathway is specifically for human cells, tissues, and cellular and tissue-based products. But an exosome preparation is acellular. It doesn't contain cells. So it lacks the foundational prerequisite to even be considered for the 361 pathway.
Oh, that is fascinating. So the clinics are pointing to the rule book for apples, trying to use it to sell oranges, and the FDA is stepping in to say, look, that rule book doesn't even apply to your product in the first place.
Yes, therefore, by default, Any exosome product intended to treat a disease or condition in humans is pushed into the 351 biologic drug pathway. They have to prove safety and efficacy in clinical trials. And the reality that every patient needs to understand is that, as of today, there are 0 FDA-approved exosome products for therapeutic use.
0. Wow. And this brings up a massive red flag highlighted in our sources regarding deceptive marketing. Because you'll see clinics advertising their exosome treatments as coming from an FDA-registered facility. And as a patient, you read that and think, oh, great, it means the treatment is safe and vetted.
It is entirely misleading. An FDA-registered facility just means a lab filled out a basic administrative form and paid a fee to be listed in a database.
So it just means the FDA knows they exist.
Exactly. It does not mean the FDA has reviewed their data, tested their product, or approved that specific exosome vial for human injection.
The liability here is just staggering. We reviewed a risk assessment from Seaport Insurance, which is a firm specializing in coverage for regenerative clinics, and they paint a really grim picture for doctors using these products.
Oh, the financial and legal risks are existential for these practices. Standard medical malpractice insurance covers doctors for complications arising from the accepted standard of care. It explicitly does not cover federal offenses like the illegal distribution of an unapproved biological drug.
So if something goes wrong,
If a patient experiences a severe adverse event from an unapproved exosome injection, and the FDA has documented cases of severe bacterial infections and immune rejections from these, the clinic's insurance policy is essentially void.
wow.
Yeah, they face federal injunctions, product seizures, and absolute personal liability.
So the U.S. regulatory environment is basically a minefield, but the science isn't confined to America. When we look at the European Medicines Agency and the UK's MHRA, they use a completely different vocabulary. Yet, they seem to be wrestling with the exact same fundamental biology.
Because the underlying science dictates the struggle everywhere. In Europe and the UK, regulators classify these complex biologicals under the umbrella of ATMPs. That stands for Advanced Therapy Medicinal Products.
Okay.
And this framework generally covers 3 specific categories: somatic cell therapy, tissue-engineered products, and gene therapy medicinal products.
Where do our cell-free extracellular vesicles land in that European framework? Because they aren't cells and they aren't engineered tissues.
This has been a huge topic of intense debate at the EMA. Currently, they dictate that native unmodified exosomes, meaning vesicles just naturally harvested from a stem cell culture, are regulated as biological medicinal products, but they're not automatically escalated to the ATMP classification.
Unless they are modified. Because the sources mention that loading an exosome with a recombinant nucleic acid changes its legal status immediately. Let's break that down. What makes a nucleic acid recombinant, and why does the EMA care so much?
Recombinant essentially means we're playing genetic architect. It's when scientists take genetic material, like a specific sequence of mRNA from multiple sources, or they synthesize it completely in a lab and recombine it to create a new genetic sequence.
Like writing custom code.
Exactly. So if a biotech company engineers an exosome to carry this recombinant mRNA payload, intending to alter a genetic sequence inside the patient's target cells, the EMA draws a hard line. The product is immediately escalated to an ATMP, specifically a gene therapy medicinal product, which brings the absolute highest level of regulatory scrutiny.
Because at that point, the exosome is no longer just a natural FedEx package. It's a highly engineered vector designed to rewrite human code.
Precisely. Now, what makes the UK's approach really interesting is how they divide their regulatory labor for all of these biological products. The oversight is actually split between 2 distinct bodies. You have the Human Tissue Authority, or HTA, and the Medicines and Healthcare Products Regulatory Agency, the MHRA.
The way the sources describe this split, it reminds me of food safety. Like the HTA acts like the agricultural inspector out at the farm. Their job is to oversee the initial procurement, testing, and storage of the raw human tissues to ensure, no infectious diseases are transmitted. But they don't regulate the final medicine.
That's a highly accurate way to visualize it. The HTA's jurisdiction ends the moment those raw cells undergo substantial processing to become a therapeutic product. Once the biological material is manipulated into a medicine, the MHRA steps in as the inspector evaluating the final dish served at the restaurant.
I love that.
Yeah, they handle the clinical trials, the manufacturing standards, and the market authorization.
But even with that clear division of labor, there's a glaring loophole that developers in both the UK and the EU are exploiting, particularly in the aesthetics market. Clinics are selling exosomes for skin rejuvenation, bypassing medicinal laws entirely by classifying them as cosmetics. How is that even possible?
It requires a clever pivot in source material and administration. So the EU cosmetics regulation explicitly bans the use of human-derived cells or tissues in cosmetics. To get around this, manufacturers extract exosomes from plants or sometimes bovine sources rather than humans.
Okay, plant exosomes.
Right. And if they package these plant exosomes in a serum and instruct clinics to only apply them topically, say rubbing them on the skin after a microneedling session, they can legally market them as cosmetics.
But there's a literal physical boundary they cannot cross because the moment the doctor takes that same legal cosmetic serum and loads it into a syringe.
The cosmetic classification instantly evaporates. The legal boundary is the tip of the needle. If a product is injected into the human body, it is classified by function as a medicinal product. If a UK clinic injects a plant exosome serum to get better anti-aging results, they are no longer providing a cosmetic service. They are committing a regulatory offense by administering an unlicensed, unauthorized medicine.
It's fascinating how a physical action changes the legal reality. So whether a legitimate biotech company is dealing with the FDA or the MHRA, navigating these pathways requires proving exactly what your product is. And this seems to be the ultimate bottleneck. How do you standardize an invisible 30-nanometer bubble? How do you prove to a regulator that batch A is identical to batch B?
Oh, this is the manufacturing nightmare of the decade. The industry calls it CMC, chemistry, manufacturing, and controls, because exosomes are highly heterogeneous.
Meaning they're all slightly different.
Exactly. Even exosomes from the exact same cell can vary slightly in size and cargo, so you need incredibly sophisticated technology to characterize them. You can't just use a normal microscope. Instead, companies rely on techniques like nanoparticle tracking analysis, or NTA.
The sources mention NTA uses lasers to track Brownian motion. I'm assuming this isn't just taking a microscopic photograph. How does tracking motion tell you the size of an exosome?
So Brownian motion refers to the random, erratic jittering of microscopic particles suspended in a liquid. It's caused by the liquid's molecules constantly colliding with them. Think of it like a massive crowd of people bumping into a few balloons.
Okay, I can picture that.
NTA shoots a laser through the liquid to illuminate the exosomes, and a camera records their movement. Because of physics, a smaller particle gets knocked around much faster and further than a larger, heavier particle.
Oh, that makes sense.
Right, so by feeding the speed of that jittering into an algorithm, the software calculates the exact size distribution of millions of vesicles in real time.
That is an incredible use of physics to solve a biological problem. But size alone doesn't prove identity, right? Because a 100-nanometer piece of dead cellular junk will jitter exactly the same way as a 100-nanometer therapeutic exosome.
Exactly, which is why the next hurdle is proving identity through surface markers. Regulators demand proof that specific proteins are present on the outside of the vesicle. For exosomes, the gold standard markers are a family of proteins called tetraspanins, specifically CD9, CD63, and CD81.
What actually is a tetraspan? Like, why those specific proteins?
Well, the name gives away its structure. Tetra meaning four, and spanin meaning to span. These are structural proteins that snake back and forth, crossing the vesicle's lipid membrane exactly 4 times.
Oh, okay.
They are deeply involved in the physical process of how the exosome buds off from the parent cell and how it binds to a target cell. Because they are fundamental to the exosome's architecture, regulators treat them like a biological license plate. If the product doesn't have CD9, CD63, or CD81, the FDA won't agree that you've manufactured a pure exosome batch.
So you've proven the size using lasers and you verified the license plates. Is that enough to get approval?
Not even close.
Of course not.
The final, and arguably most difficult hurdle, is potency. You have to prove the FedEx package actually delivers a working message. Companies must validate a biological assay, which is a repeatable laboratory test that demonstrates their specific batch of exosomes consistently performs its intended biological function.
Give me an example of that.
So if your clinical claim is that your exosomes suppress immune responses, you must have a standardized bioassay proving that when you apply batch after batch to immune cells in a lab, they reliably and consistently dial down inflammatory markers.
And considering a stem cell might alter the cargo of its exosomes based on, I don't know, whether it was stressed or what nutrients it was fed that week, guaranteeing that identical potency across thousands of vials must be astronomically difficult.
It is the single highest wall-blocking commercial exosome therapies right now. The underlying science shows immense promise, but demonstrating the rigorous manufacturing consistency demanded by the FDA and EMA is a threshold the industry is still really struggling to cross.
We have unpacked a tremendous amount of complex science and law today. We mapped the biological divide between living cellular factories and cell-free extracellular vesicles. We decoded the U.S. regulatory maze, showing exactly why culturing tissue triggers the rigorous 351 biologic pathway and why the FDA unequivocally rejects the idea of unregulated exosome therapies. We explored the European ATMP framework and the UK's division of labor between the HTA and MHRA. And finally, we looked at the sheer physics required to manufacture and measure a 30-nanometer therapy.
It's a lot, but the overarching theme is that regulatory bodies are working aggressively to ensure that the promise of regenerative medicine doesn't outpace fundamental patient safety.
And for you listening, this knowledge is your armor. If you are ever evaluating a regenerative clinic, you now know the vital questions to ask. Don't be swayed by sleek marketing. Ask if the cells were expanded in a lab. Ask if the product has actual FDA approval, not just a facility registration. Ask if the cosmetic serum they're holding is legally allowed to be injected. You have the tools to navigate through the jargon.
And those questions are really necessary because the regulatory landscape is only going to get more complicated from here.
Which leaves us with one final provocative thought. Throughout this deep dive, we have discussed how governments regulate natural extracellular vesicles. Products ultimately harvested from living cells, whether human, plant, or bovine. But the field of synthetic biology is moving at breakneck speed. What happens when we don't need a biological factory at all? When biotech companies start using nanotechnology to build artificial exosomes from scratch, constructing custom lipid bubbles and loading them with synthetic RNA.
It's a fascinating dilemma. If our entire regulatory framework for tissues and cells is based on the premise of biological origin and minimal manipulation, how do agencies like the FDA or MHRA classify a biological FedEx package that was built by a machine completely devoid of any biological parent cell?
It is a profound regulatory blind spot, and it is hurtling toward us faster than we think. Thank you for joining us on this deep dive. Keep questioning the science and the systems that govern it.